Hello, I'm Jonas Nielsen from the University of Michigan. First of all, thank you, Dennis, for inviting me to present our work at this webinar or journal club. Today, I'm going to present our most recent work on genetics of atrial fibrillation. But first of all, I would like to emphasize that the work I'm going to present reflects a large collaborative effort with contributions from collaborators from all over the world, including Iceland, Norway, Japan, Denmark, and several places in the United States. As you might know, atrial fibrillation is the most common cardiac arrhythmia with huge clinical implications and high cost to the society. Despite this, our understanding of the pathophysiology underlying this cardiac arrhythmia is still relatively limited, and current treatment regimes are not always very effective. At least they are very rarely curative, and they can have severe side effects and complications. The aim of this study was to learn more about the underlying biology of atrial fibrillation, to highlight potential new therapeutic targets, and we aim to do this through large genetic and genomic analysis. Today, I will only highlight our most important findings, but if you're interested in more details, you can always read our paper preprint on bioRxiv. So before I'm going to present our findings, I would like to give those of you who are not familiar with atrial fibrillation a very brief introduction. During normal sinus rhythm, each heartbeat is initiated in the sinus node, which is located here in the upper right corner of the right atrium. From the sinus node, the electrical impulse travels further down to the AV node, and from here further down in the ventricles of the heart. And altogether, this will trigger cardiac contraction and hence make the heart work as a pump and circulate the blood throughout the body. On the surface EKG, you will see these regular waves. In case of atrial fibrillation, the normal coordinated electrical impulses in the atria are replaced by small chaotic electrical waves traveling around more or less randomly. So this will give rise to an irregular and often fast heart rate. And on the EKG, you will see that the normal P waves representing the contraction of the atria are replaced by small fibrillatory waves representing the chaotic electrical activity in the atria. There are many risk factors for atrial fibrillation. The most important ones include age and male gender, but we also know that, for instance, valvular heart disease is a huge risk factor. As you can see here in this echocardiography, where you see that the mitral valve is leaky, so the blood is going in the wrong direction, and then that can over time leads to pressure overload and dilatation of the atria, and this is an important risk factor for developing atrial fibrillation. Other important risk factors include ischemic heart disease, hypertension, also various metabolic disturbances, for instance, like diabetes and hyperthyroidism. But we also know that atrial fibrillation has an important genetic component. It has been shown in twin studies that atrial fibrillation has a total heritability of around 60%. And also previously there has been identified a number of regions in the genome associated with atrial fibrillation through genome-wide association analysis. Atrial fibrillation have important clinical implications. Most of all, is the most frequent cardiac arrhythmia. I'm sorry to tell you that 1 in 4 of you will likely develop atrial fibrillation during your lifetime. And currently, atrial fibrillation affects more than 30 million people worldwide. Atrial fibrillation can be associated with palpitations and shortness of breath and decreased fitness, along with heart failure. But most importantly, atrial fibrillation is associated with a high risk of stroke, and this is because the fibrillating atria can give rise to blood clot formation, and these blood clots can go loose and go to the brain, where they can occlude an artery and cause an infraction and hence a stroke. Current treatment strategies include medications that either control the heart rate or the heart rhythm. However, it's also possible to try to go up in the left atria with a catheter and try to burn or freeze the areas with chaotic electrical activity. Most often, you would try to isolate the four pulmonary veins, because atrial fibrillation is often triggered by ectopic impulses coming from these veins. However, this type of ablation treatment can lead to severe complication, and usually atrial fibrillation will reoccur after some time. So we performed a large genome-wide association study, meta-analysis, to improve our understanding of the genetic variation underlying atrial fibrillation. And this was with the hope of improving future treatments and patient care. So we compared a total of 60,000 cases with almost 1 million controls of mainly European ancestry from 6 contributing studies, including the AF Gen Consortium, which has kindly released summer statistics from the most recent GWAS. So across the 6 contributing studies, we were able to analyze a total of 34 million genetic variants scattered throughout the genome. So here you see a classical Manhattan plot summarizing the findings. On the x-axis, you have the genomic position by chromosome, and on the y-axis, you have the minus log 10 p-value of the association for each genetic marker, each represented as a dot on this plot. So it might be hard to count on this plot, but we identified a total of 111 independent genomic regions, or loci, associated with atrial fibrillation. And of these, 80 have not previously been reported. The novel findings are indicated in red, whereas the known regions are indicated in orange. So the most statistically significant signal we saw was for the PRITX2 locus, which is also the first locus that was reported for atrial fibrillation. I think it was back in 2007. And for this, the top mark on this locus, we observed a p-value of 7 times 10 to minus 443. So identification of genomic regions associated with atrial fibrillation is of course nice, but it will really not add anything to our understanding of atrial fibrillation unless we try to understand the underlying biology of these associations. So in order to do this, we continued and performed a number of downstream analysis that I will just walk you through. First, we use data from almost 40,000 human mRNA microarrays to identify tissues and cell types in which genes from atrial fibrillation, from the atrial fibrillation loci that we identified, where genes are preferentially expressed. And so on this plot, on the x-axis, you have the x-axis represents the 209 tissues and cell types that we tested, whereas the y-axis represents the minus log 10 p-value for the tissue-specific expression. And the red columns represent statistically significant tissues followed Bonferroni correction. So reassuringly, we found that genes that fall within these 111 loci that we identified were preferentially expressed in tissues from atria, atrial appendages, heart, and heart ventricles. So Next, we performed a pathway analysis based on 15,000 predefined gene sets that were reconstituted based on gene expression experiments. By doing this, we were able to identify almost 900 gene sets that were enriched for genes located within these 111 atrial fibrillation associated loci that we identified. On this plot, I just showed you the top 28 gene sets. I couldn't fit in all of them. The gray lines between the gene sets just represents the overlap of genes across the various gene sets. And as you might see, the highlighted gene sets, or the top gene sets for atrial fibrillation, in general point to biological processes related to cardiac development and morphology, along with structural remodeling of the myocardium, including, for instance, failure of heart looping, thick ventricular wall, or atrial septal defect. So we found that most of the atrial fibrillation associated risk variants that we identified were situated in the non-coding part of the genome. This might indicate that many of the risk variants that we found to be associated with atrial fibrillation might act via, for example, regulation of gene expression or correct timing of the gene expression rather than affecting the structure of the proteins themselves. So to explore this further, we downloaded data from the roadmap dataset, which holds genome-wide information on various regulatory regions, including, for instance, data on open chromatin, which indicates regions where the DNA is accessible and open for gene transcription. Using these data, we tested for an overlap between the various regulatory regions and the identified atrial fibrillation risk bands. And we did this using our own software, Gregor. So this plot shows the results from this enrichment analysis. On the x-axis, you have the fold enrichment, and on the y-axis, you have the p-value for the enrichment. And out of the 800 combinations of regulatory features and tissues that we examined, each indicated by a plot, or by a dot in this plot, we found that the atrial fibrillation associated variants were in general more strongly enriched for active enhancers, for instance, indicated by H3K27 AC, which is a histone marker indicating active enhancer activity. And we found the strongest signal for right atrium and left ventricle. But interestingly, we also found enrichment of DNA hypersensitivity sites reflecting open chromatin in the fetal heart and tissues from fetal heart. So we think that this suggests that some of the loci we identified are important for transcriptional regulation in adult heart. Some are important for transcriptional regulation in the fetal heart, or it could be both. To further enhance our biological understanding of the atrial fibrillation associated loci we identified, we tried to identify functional candidate genes likely to be involved in atrial fibrillation. We found that within these 111 loci that we identified, we found that there were, of course, it's a bit arbitrary how do you define your loci, and you can see the details in our paper, but based on the definitions we used, we found that there were a total of 3,072 genes within these 111 loci. So we tried to prioritize the genes, and we prioritized the gene if it was the nearest gene to the locus lead variant, if the gene harbored a protein altering variant, or if it was an LD, high LD with a variant that was altering the protein. We also highlighted a gene if the locus lead variant was an EQTLS for a gene, meaning that the variant affects the expression of the gene, or whether, or also we included the genes if any of the top variants LD bodies was associated with gene expression of a gene. And finally, we included genes if they were highlighted in our pathway analysis. And then we were able to filter these 3,000 genes down to 162 candidate genes for atrial fibrillation. So we tried to evaluate this finding. So based on the GTEx gene expression dataset, we found that the 162 genes that we prioritized were preferentially expressed in heart muscle tissue, and also in blood vessel. So we tested the empirical significance of these results by performing a number of permutations, each indicated by a gray dot in the plot here, and on the x-axis you have all the different tissues that we tested. So first, we randomly sampled 162 genes from the entire genome, and this was repeated 1,000 times. And then the blue line here indicates the 95th percentile of these 1,000 p-values for each tissue. And then we repeated the experiment, but instead of sampling genes from the entire genome, we sampled genes from within these 111 loci. And again, we performed 1,000 permutations, and the 95th percentiles of the p-values are indicated by this yellow line here. So altogether, we found that the observed p-values for the 162 genes that we prioritized were substantially more significantly expressed in heart, muscle tissue, and blood vessel tissue than compared to expected by chance. So we believe that these findings support that the genes that we prioritized are actually strong candidate genes for atrial fibrillation. So previously, or at least some years ago, when people were discussing the genetic substrate for atrial fibrillation, they were mainly talking about ion channel genes, so-called channelopathies, and how mutation in such ion channels could affect, for instance, the cardiac action potential, and then ultimately lead to atrial fibrillation. So these discussions were mainly based on a number of family studies which have identified rare mutations in ion channels that segregates with atrial fibrillation in larger families. So among the 162 genes that we prioritized, we reassuringly also identified a number of ion channel related genes, and also a number of genes involved in calcium handling in the heart. However, we identified even more genes known to be involved in cardiac and skeletal muscle function, including, for instance, the SGCA and SSPN, for which coding variants have been associated with muscular dystrophies, and also the PKP2, which has been associated with arrhythmogenic right ventricular cardiomyopathy. So we also found several genes that has been previously associated with congenital heart defects in human or mice. We've identified a number of genes known to be transcription factors and involved in, for instance, cardiac development or development of other organs. We also identified genes known to be involved in angiogenesis, and also genes involved in hormone signaling in the thyroid receptor. We also performed an so-called ECG-wide association analysis. So basically, we just took the 111 index variants and we tested them for association with 123 EKG trials, and this was done in 60,000 people from Iceland. And by doing this, we found that 60 of these 111 variants were associated with at least one EKG parameter, and 39 of these were novel in atrial fibrillation. So finally, we constructed a polygenic risk score using weighted effect estimates generated from the large Icelandic sample, and then we tested the performance of predicting atrial fibrillation in another of the larger studies that we had access to, the Hunt study from Norway. So we basically generated a genetic risk score. We basically generated multiple genetic risk scores across different p-value thresholds and LD thresholds, and we found that variance, that if we used a p-value threshold of 5 times 10 to minus 5, and if we used LD pruning using an R-squared threshold of 0.8, we got the strongest risk score for atrial fibrillation when we tested in this independent Norwegian cohort. So we then used this optimized polygenic risk score to test for association with 1,494 disease-specific trials in the UK Biobank. And we used, for this analysis, we only used people of white British ancestry. So in addition to a strong association with atrial fibrillation here indicated with a p-value of 7 times 10 to minus 374, we also found a strong association with 33 mainly cardiovascular conditions, including, for instance, palpitations, mitral valve disorders, hypertension, heart failure, ischemic heart disease, and stroke. However, when participants diagnosed with any type of cardiac arrhythmia were excluded from the analysis to avoid, for instance, assertment bias, then the risk score was not associated with any ICD-specific disease group. So we believe this suggests that the score we generated is specific for atrial fibrillation or cardiac arrhythmia in general, and that the additional associations that we identified were mediated through atrial fibrillation, either as a result of a more thorough clinical examination. For instance, you could imagine that a person with atrial fibrillation more often is referred for an echocardiography, and then you will discover that they have a valvular heart disease, for instance, or it could also be because that atrial fibrillation is a likely intermediate step towards the disease, for instance, in the case of stroke. So in the end, I'll just summarize our findings. So we performed a large genome-wide association study, meta-analysis of atrial fibrillation. We included a total of 1 million people and 34 million genetic variants. Doing this, we identified 111 loci, of which 80 have not previously been described. We also prioritized a total of 162 atrial fibrillation functional genes, and many of these genes could potentially represent novel drug targets. We have also done some gene-drug interaction analysis that you can read more about in the paper, and we highlight some potential drugs. Based on pathway and functional enrichment analysis, along with prioritization of functional candidate genes, we anticipate that many atrial fibrillation risk variants act in the developing heart or impact atrial fibrillation via structural remodeling later on in life. These findings, of course, needs to be confirmed but we believe they provide a strong foundation for directing future functional experiments to better understand the biology underlying atrial fibrillation. So just to put this a little bit into perspective, like previously genetic predisposition to atrial fibrillation was previously thought of as a sort of as defects in cardiac ion channels. Our results, along with other number of other recent reports, indicates that perhaps the genetic predisposition to atrial fibrillation acts via muscle structure abnormalities, or during the fetal heart, during the development of the fetal heart, and perhaps we should start thinking of atrial fibrillation as a atrial cardiomyopathy with a heritable component, like in the same way as we have the, for instance, dilated cardiomyopathy or arrhythmogenic right ventricular cardiomyopathy, which are also associated with a high risk of, for instance, ventricular arrhythmias. So finally, just say thanks to all the people who contributed to this study. It's people from all over the world. Especially, I would like to thank my mentor, Kristen Willer. Thank you very much for your attention.